Soil is its own living, dynamic ecosystem composed of mineral and organic solids, gases, liquids, and living organisms, which is capable of supporting plant growth. An acre of soil can hold 5 to 10 tons of living microorganisms. 1 gram of soil can hold as many as 5 to 7,000 bacteria species and more than 1 billion individual microbes. The bacteria and other microorganisms are necessary to break down dead and decaying matter and turn it into usable energy by living plants. Soil has several essential roles. Healthy soil holds more water, reducing runoff and evaporation. The productivity of all ecosystems, whether agricultural or natural, depends on soil health. Soil minerals act as a filter, while soil microbes degrade and detoxify a variety of materials, including some industrial waste products. Nutrients are stored and cycled through the soil. Finally, soil structure provides a scaffold for plant roots and support for our houses and roads. In our research, the third critical control point examines the role soil plays in the fate of antibiotics, antibiotic-resistant bacteria, and antibiotic resistance genes. Does the kind of soil to which you apply the manure or compost matter? In a bench-scale microcosm study conducted by Dr. Gia's team, compost samples from the previous composted manure study were amended to 3 different soil types and incubated for 120 days. Samples were collected at specific intervals during this period and stored. In a greenhouse study, lettuce, broccoli, and radish seedlings were grown in loam or sandy soil with 4 treatments: compost with antibiotics, compost without antibiotics, raw manure with antibiotics, and a control with chemical fertilizer. After growing for 3 months, soil samples were collected, examined, and analyzed. Finally, the field study conducted at Virginia Tech's Urban Horticulture Center entailed growing lettuce and broccoli in the same loam soil as the greenhouse study, with plots for each of the same treatments. Samples of the soil were taken from these replicate plots and analyzed. Stormwater runoff from each plot was also collected and analyzed for sediment, indicator bacteria, and antibiotic resistance genes to determine whether these were moving downstream to other fields. State-of-the-art tools were used for each of the studies to determine the fate of antibiotics, antibiotic-resistant bacteria, and antibiotic resistance genes. This study will provide guidance on whether certain soil types amended with composted manure have an effect on the transfer of antibiotic resistance.